So okay, just to get you up to speed on how I came to where I was, is 91 to 95. I did my bachelor's in chemical engineering at Indian Institute of Bombay. I came to Michigan, had a fellowship, and then uh, I was lucky enough to get a fellowship to not get assigned to any research group early on. So I got a chance to really explore what's going on at the department. And at the time, again, I had to make a decision. I mean, I got into the fellowship in the PhD program, but I was really thinking at the time, should I do my PhD or not? Because I was pretty sure that I will get to go to industry at some point. So very early on, I was actually talking to a lot of people and trying to figure out, should I get into this 5-6 year PhD track, because at the end I'm going to go to industry anyway. So along the way, as I was talking to people, Mark gave a presentation on the project he was doing on microfabrication for DNA analysis. I was hooked. I said, I'm going to do this project, and if I don't get it, I'm going to quit. And that's what I said in my, in my selection. I gave Mark as the only project I want to do. And lo and behold, I did get the project. And after that, pretty much, I became a lab rat. I was fortunate enough to be in a very highly interdisciplinary group. Thanks to the 3 advisors, we had people from electrical engineering, human genetics, chemical engineering. So I think I got a taste of what it would be like in industry very early on. You talk, use different disciplines. The biologist pose the problem, the electrical engineers, chemical engineers try to come up with solutions. And I think I got a chance to see what it takes to solve a problem, that will have an end result that could be used by someone. And along the way, I think I got a chance, thanks to Mark, I got a chance to go, attend conferences early on. That was my 2nd year. And that's when I think I got the startup bug. I was noticing some of the best excitement, the best results were coming from the startups. You can see that their thinking is different from the academia. And I think I started to conceptualize my own career path early on, that hey, at some point, once I go through my the thesis defense, I'm going to do a startup, because it's not enough to create new technologies. To get it to a product, it takes a lot more work. And I think I would like to have that experience of customer using what I have created and say, hey, I think this solves my problem. So I knew I was going to do a startup, so I made my intention clear. So we had really a fun ride in the whole team at Michigan. We created unique technologies. We had that article in Science that gave us notoriety. And I was pretty ready to move on. In '99, I was trying to figure out what to do, and I was actually interviewing startups across the country. I had good offers, but coincidentally, some local investors actually came up to Tech Transfer, talked to David, talked to Mark, and they said, hey, you guys have created some unique technology. Would you like to commercialize it? And that's when I started, thinking about, switching my career path from working for a startup to creating a startup. So I'm getting ahead of myself. So this is the science device that Michigan is quite famous for. So that gave us a notoriety. And then so '99 is when I first met with the local investor. Again, before meeting the investors, I had to learn quickly, is this the right thing to do? Because, I've been used to being in the lab. I only know technology. I mean, I have no clue, about business, accounting, market research. All those terms started, appearing in the communications. So I had to quickly learn whether, I should go ahead and meet these investors or not. And, you go through these moments of, reflecting and thinking what to do. And I reached out to people that are my friends that may have been associated with startups and other, aspects of, commercialization. There was one friend of mine from my IIT Bombay days. He had just done a startup in the, .com area and had just going into Harvard Business School. So he said one thing, every day in a startup is a learning experience. No matter what happens, If you go through it, at the end, you will come out so much knowledgeable and experienced. You will not get that anywhere else in a big company or in academia. So that kind of stick on to me, and then I pretty much had to evaluate my risk at the time. I was a bachelor's. I was living on a student stipend. I think it was about $1,200, and then I told myself there's nothing to really worry about. If I do a startup, I'll get less pay as whatever, it'll be less amount of money, but it'll be bigger, it'll be more than what I get paid as a student. I don't have a family. All I need is doing the work that I like and get 8 hours of sleep. And then everything else is a plus. So I said, okay, I'm going to jump onto it. We decided to meet the investor. And I think it's quickly learning about the different aspects of commercialization. I think we hit it off well. I came back to the group, asked other people in the team. Sundu, who was my just a few months or maybe a year behind graduation, he said, hey, if you do it, I'll join you. And pretty much we started going out and trying to figure things out, how to get the technology out from university and create a startup. So we're lucky enough to get a chance quickly in the sense that we had the VCs interested. They were willing to work with us. So what we did was we worked with tech transfer. We worked with tech transfer and the investors, and we came up with a game plan. The VCs would give us $75,000 if we can get the IP license from University of Michigan, get IP lawyers, vet the IP landscape, and figure out if there's a space for the new company to operate, and then essentially incorporate the company. So that's what we did for 6 months. We presented to the investors. That's the vision we came up with. Palm was hot at the time. We said we'll create a handheld laboratory for doing DNA analysis. That's the area that we knew. And then in early 2000, we incorporated the company. Along the way, we got the UFM license. We got the IP review from lawyers. Came back good. And then we got about 2.4 million Series A. I think it was May 18th. So about 15 years back now, I defended my thesis. May 19, 2000, we started at one of the investors' open room and started our operations. So again, it's a fairy tale kind of story, but a lot had gone into the early days. Again, I think we are lucky enough to get that 75,000 and good relationship with the VCs to be able to start a company. But if I look back now, this is an awfully wrong way to get a company started, because there are a lot of unknowns. And now that I know how difficult it is to make money in commercialization, that $75,000 is a lot of money for what the risks were. We had 2 PhDs, knows about the technology, no idea how to commercialize, no prior history. And then there's a lot of unknowns on the commercialization. It's a new technology. So I think EDF Ventures took a huge risk on putting that 75,000 and then eventually 2.4 million. I think the times were different. That was a time where .com was really booming. A lot of investments were getting poured into new startups, and I think we're lucky to be at the right time to get a couple of $1 million to start the company. So that was the goal. If you see the handheld, that's what we started out. But as we started learning more about ourselves and the problem we were trying to solve, this is what we came up with. It took 6 years to get there. A box, it's not a handheld. It need not be a handheld for what we were trying to do at that time. And there was a couple of disposables. The key product is a microfluidic disposable. You put a clinical sample, put the cassette in the machine, shut the lid, and it'll give you a result on looking for certain DNA of bacteria so that you can predict some infections in 1 hour. So it worked like we had wanted it to, and having really cutting-edge microfluidic technology behind it. Again, 6 years it took us to get to that, because there were a lot of new technologies we had to create to get this to work. And then you might think, okay, this would work. And the first area that we used the product was for group B strep infections that was to be detected in pregnant mom. It was critical to detect the bacteria in time. And then, so it had all the right attributes to be a successful product, but we still decided to not go ahead into the market with this. There are a couple of reasons. Even though we wanted this right next to the patient in hospital delivery rooms, the hospitals were not willing to put it next to there. They would still put it in the centralized testing lab, and there with this product really didn't make sense because you would have to run at least 50 samples a day. You cannot have 50 machines sitting there, in that hospital testing room. So we went back to the drawing board, and then we told our investors we'd already burned through about $30 million to get to that, and said, hey, the first product is not going to cut it. We need another chance. And it was a huge turmoil first to get all the investors on board. And then also the team, the company was probably up to about 40 people at the time. So it was definitely a struggle, difficult change to go through. But we got one chance to get it right, to be in sync with the market, and this is what we built in about 18 months after that. It's an automated machine that combined robotics, liquid handling robotics, and microfluidics. It can do 24 samples at the same time, 24 results in about 1 hour. The first result in 1 hour, 24 results in about 2.5 hours. So it had all the attributes that the market needed, and this was immensely successful in the marketplace. So I will try to go through, the timeline, what happened. We started in 2000. In 2006, we created the, point-of-care product. It took about, 3 years after we had created all the core technology. But we shelved that. The hospital product that we, launched in 2007, 2008 was immensely successful. We had, a lot of interest from all the hospitals. They started buying the machine. And then we saw a lot of interest from corporate partners like QIAGEN, Becton Dickinson, other companies like that. So we had a deal with Becton Dickinson in 2009, early 2009, and by the end of that year, we actually got acquired because there was a lot of competitive pressures on the company by other companies to be bought out, and BD essentially to protect some of their investment had to buy us out. So if you see, right from starting out as a startup, 2000, it took us 9 years to get it right. And along the way, about 6-7 years of, I would say, really nurturing the technology to be able to create something unique. This is something you do not get a chance nowadays to get so much incubation from investors, upfront, 4 to 5 years or more of just developing technology. So that pretty much has to happen at the universities or some settings where you would have to do the development, the technology development in-house, and not have to do it at the company. So again, it's a snapshot of a lot of things that happen. But if you see, we did learn a lot along the way, working as a team. I think we were up to about 70 people when we got acquired, and then that 2009 is when we essentially became part of BD. So I was with BD for about 15 months. So we are working on the next generation product. So I helped get that to the FDA trial. I was busy for about 9 months, 10 months, and then I started getting really bored at a bigger company because the pace of activity is a lot different. You're probably talking 10 to 20 times difference in pace of what you expect to see in a startup versus a big company. So that started really getting to me, and then I was yearning for a change, and that's when I started looking around, and then eventually jumped into de novo sciences. So there's a story behind that too. I'll tell you some of the what happened, how we got de novo sciences started. So again, HandyLab was successful for a couple of reasons. I think the core team, myself, Sundu, and some of the key people early on, at least the first 7 to 10 people, are all very innovative people that really likes to make a difference. I had a couple of students from or engineers from Mark's group, Gene, Aaron. They're all doing well now. They were all part of the de novo team, HandyLab team early on. So if you look back at the key people that joined early on, I think they made a huge difference in setting the right culture at the company, and that was key in making the company successful. And then the other thing that we did as a team was we stayed focused on milestones. We had very good milestones along the way, and then if you don't meet them, you pretty much will find it very hard to go ahead to the next step. The team was very committed, hardworking, and then we also were very honest about our own capabilities and always asked for help when needed. And I think the team worked good together and really had fun, and I think that was also very important. Again, those are all good things, but eventually HandyLab was successful because of the business reasons. First of all, it had a very rapid, sustainable revenue growth. We made an instrument, but there are hundreds of applications or assays you can put on the machine, and then there was great margins for the company. You are talking about 70% gross margins on the product, which made it very attractive for us being acquired, and then also very highly efficient operating costs. We were very efficient in how we manage our people, so that kept the company very lucrative for acquirers. So I think this is why you get bought, not because you have a nice people in the company and you have fun while working. Just to summarize, some of the key ingredients of a startup success, team and culture, having the right team and having the right culture in place is very important. Again, you have to be developing a product that solves a need in the marketplace. It cannot be a me too. It has to be really solving a problem that's really difficult, causing pains for the customers. For any technology company, you need a lot of dollars for developing the product, patience, and I think also luck. Luck to get the funding that you need along the way, and also having the right opportunities available along the way. Again, I learned a lot through that experience, and then you cannot get that being on the sideline or outside of it. You have to be in it to really learn from it. Okay, I'm going to switch gears to my new startup called de novo Sciences. You will see how it'll come together, some of the basic attributes of the two startups. So this is my new startup. It's called de novo Sciences. The first startup was in microfluidic infectious disease detection using automation and integration. This is in cancer diagnostics. We are looking at cancer monitoring and therapy selection assays. This is the product we have launched about 4 months back. There are two machines. One will do sample prep. One will do detection, imaging and detection. So it will essentially take blood, take it through the system, and provide information on the metastasis process, characterization of the cancer cells, and it'll do from blood, which is a big deal because the current gold standard for doing cancer detection is through tissue biopsy. You essentially do surgery, get a chunk of the tissue, and analyze for different markers. So this will make it a lot more easier for the patients. It'll also be more relevant because as cancer spreads from primary tumor to different parts of the body, you actually you have to biopsy different locations. Sometimes you don't have any materials to surgically remove and test. So having a blood test is it's a huge deal, and I think this will be the first system ever in the marketplace to provide you that capability. So again, I like to go back to my core, which is microfluidics. So we essentially have a microfluidic device that captures cells. The slide that captures cancer cells from blood has 56,000 capture wells. There are new generations also working on 100,000, 1 million wells, things like that. So we capture cells and then look at either protein markers, antibodies, or look at mRNA through fluorescent in situ hybridization, DNA mutations. We can grow cells. or you can take out the cells and do either PCR or next-gen sequencing. Again, we're trying to replace tissue biopsy, so any tests that you do on a tissue slide can be done by from blood using our slide. Just a summary of this company. So it's a differentiated platform, fully automated, system for liquid biopsy, unmatched, proprietary microfluidic chip technology, and we are initially focused on metastatic prostate, lung, and breast, and their therapy selection assays. And our business model is very similar to my last company. We'll make the instrument, have it be available for cancer centers, reference labs, CROs, and eventually hospitals once we get FDA approval, and then generate revenues from the disposables, high-value disposables. So as far as commercial status, we launched the system end of last year for research use and clinical validation. We sold 2 instruments. Surprisingly, those were purchased by not within the US, but one in India, one in Qatar, and then there are a few other countries very active. What we are seeing is there's a huge vacuum of technology need outside United States, and they are clamoring to get some solution. So we are finding a lot of demand outside the United States. There is a demand here too, but I think there are a lot of options available to customers here. And then we are engaged with FDA for a clinical trial for the first assay. So eventually we'll have this product be cleared for monitoring and prognosis of prostate cancer first. We hope to get all the trial done by next year, and then we can start really selling it to the hospitals. Again, this company have raised about 5.67 million to date. Again, a lot more judicious with the money. We got our first bolus of funding in end of 2011, and then by end of 2014, we have a full automated system launched. That's something very one of the industry first in the world to have a completely automated system like that developed and be sold in the marketplace with less than $4 million burnt. I think I have the benefit of going to the first company, and I also have the same key people helping me out, build this company out. So, again, we've done an awful lot with a little bit of money, and we hope to really make a difference. Okay, it's not all easy how de novo sciences started, so I'm going back to. So right when I was with Becton Dickinson, early 2000, late 2009, early 2010, my wife, Priya, she's a student of molecular biology. She used to, was interning at another startup called Aspirin Therapeutics. I'm sure you probably know Dr. Roger Newton. He's a very well-known person in the cardiovascular space, co-inventor of Lipitor. He runs Aspirin Therapeutics, another startup. So Priya used to work for Roger, and then Chris was another colleague. And startups go through an awful lot of changes quickly. They sometimes when the money runs out, or it's getting low, you have to really let go of people. So Priya and Chris were one of those 2 people that was given the pink slip. And then I was out. Sorry. I was out in Quebec City training some of the manufacturing people at BD Quebec on the HandyLab. They're going to move manufacturing of HandyLab to, or BD at the time, to Quebec. So I was out there, and then my wife gets she gets a pink slip out of job. She's on the phone with me. She's crying in a couple of hours, and I told her, Priya, you don't need a job. We're okay. We're okay. You don't need to. That was 2010. Height of recession. It's hard to find a job. If you have something, you hold on to it. And I told her, you don't need a job. I mean, you can do something on your own. And that something clicked in her mind. And next morning, she's out talking to her friend Chris, and also another fellow molecular biologist, Saida. Priya and Saida did their masters at Eastern molecular cell biology, and Chris was another biomedical, not an engineer, but he's a biomedical student from Grand Valley. They all happened to be in Ann Arbor town, and they knew each other. So they got together, and Priya said, I'm going to put whatever money I got from my internship into the company and start a company. We'll figure out what to do next. I said, go ahead. I mean, there is no risk from my point of view. Hey, I think because of the HandyLab exit, Financially, we were very stable. I mean, there was no issue about we could not work for some time and still be okay. So it was a simple knee-jerk reaction to her not having a job, and said, hey, you can do that. And then next morning they talked to lawyers, incorporated a company, and then they essentially started figuring out what to do. I mean, they had degree in biology, so started a whole list of ideas, screening through them and trying to figure out where they could commercialize something, either sell somebody else's product, get it from China, India, and sell it here, things like that. So they came up with a whole list of ideas. And my role at that point was to just give them the next milestone. That's what I did. I told them, you incorporate a company and you figure out where you can create a business. That's what I told. So they are out doing their own stuff, trying to figure out. And then they also thought, because they were a student of biology, they could apply for some grants. And then GATE Challenge, on the face of it, they make it look very easy for anyone to apply for a grant. So they actually applied for one of the grant challenge. And what it did was it brought them together to think on the same project. So I applied for it. They were very motivated about what their ideas, and they were eagerly waiting. Okay, we're gonna get this grant, and there'll be things will be all hunky-dory after that. So it was a couple of months of waiting, and then along the way they did get into some boot camp, entrepreneur boot camp. First of all, being from a small university in a big university town is awfully hard on them. Everybody else would tell them, you guys don't have a PhD, just a master's student from Eastern, just go get a job. I mean, this is not your, not your forte. So, some of the people's comments were very, I would say, very insensitive. And I think it was important for the team to use that as actually something, as a personal challenge. Hey, I'm gonna show it, that kind of thing. So, they would all be upset every once in a while, and I'll just say, hey, doesn't matter what people say, you do what you need to do at that time. So, and September came, they got rejected from the GATE Challenge. Obviously, it's a very competitive, funding process. Now they had to figure out a real business, not just write a grant. So they used their GATE Challenge, ideas, and started creating business, because the GATE Challenge proposal was more for infectious disease in developing countries. Nobody will fund you for that business in US. So, my advice to them was try to turn it around to a problem that needs to be solved in the United States. And that's what they worked on, for a couple of months. And then along the way, they saw that there was going to be a huge competition in Ann Arbor called Excel Michigan. It was, it brings in companies from across the world to compete for about $500,000, and there are a lot of business plans being presented. So what the team did was they didn't have money to, they're all surviving on a little bit of money that was invested by Priya. They didn't even have health insurance, so they tried to be very careful with their lives, not to get hit or whatever. And then it was funny, we didn't have an office. They all worked out of my home. Priya would cook lunch for them to save money for the other two. So it was really working together day in and out together on the same problem. So one of the opportunity came is they wanted to attend all the business plan competitions at Excel Michigan. Again, it was $350 to get in. So they didn't want to spend the money. They said, hey, can we volunteer and help out? So went in as volunteers. One held the mic. One would be pointing the crowd towards certain venues. So what happened is they sat through probably 30 to 40 business plan pitches, different formats, 15-minute talk about describing your business. And then at the event, they met probably 10 to 15 successful entrepreneurs and talked to them about their own ideas. And when you talk to people and they ask about what you do, and when you have to answer, you really have to have a good, compelling answer for what you do. I think at the end of that event, they realized that their business plan sucked. they had nothing. Nobody would even put money, and I think that realization came, and it's like, hey, we don't have much money left. Either we disband and go our separate ways, and then or figure something out. So I think those those events were very important. So the other thing that happened along the way is they knew that they had to revamp the business plan. And Priya and I had a friend in Chicago. He was going through some trouble in his bowels, so he was getting all kinds of checkup. And then that was around Christmas time. And his dad was going through the same kind of situation in India. And while we were there, visiting them, they realized that his dad has pancreatic cancer, has only a few days. And the son, my cousin, or Priya's cousin, was trying to figure out his own problem, and then they're kind of completely clueless what to do. I mean, he himself has problems, and now he has to figure out how to get to India to see his dad. And then there were a couple of days where there was crying in the house, and then we were just supporting them. And Nandini, who's the wife of my our cousin, she asked Priya, hey, you are a student of biology, don't you have a definite answer whether there's a solution for all this? Some of those questions really hit you, and then it really forces you to think about, hey, why am I not trying to solve this problem? So we actually put them in the flight to go back to India. Dabu, which is our cousin, his dad expired within a few days. Dabu was lucky to see him for maybe the last few hours. And then driving back, Priya and I, were long dead silence. We were all reflecting what to do. And then we came back, and then Priya said, I want to do something in cancer. And that's when she got her team looking into the problems in cancer, how to help figure out some technological solutions for that. So lo and behold, once you dig in, there's a huge unmet need in cancer. Again, it's all tissue-based diagnostics. You're looking for a blood-based detection. And then what they did was they talked to, emailed. They already had some biological idea how to approach the problem by doing lit search, scanning through all the available technologies. And they started talking to, emailing cancer researchers, oncologists at U of M and Karmanos. And then within weeks, they had met probably about 10 of them. They all said it's a huge problem, and this is what's needed. And what they did was the team started working with some local business plan experts, Michigan Small Business Technology Development Corporation. And then they were able to put together a business plan. And then in June, they competed for this business plan. They got the best new business idea. It gave only $5,000. And then Priya won the best elevator pitch award. And that's when I thought, hey, this is for real. I need to jump in. And yeah, because all along I thought, hey, this is just going to go away. I'll become. Actually, honestly, I was at BD at the time. I knew I will, jump from BD because I was getting very, very bored at BD at the time. I was working with few other startup entrepreneurs, also a couple of local VCs, either to become a VC, full-time venture capitalist, or become management for another startup. But once Priya and her team won the best new business idea, and then when your wife says, hey, you need to help me, then you go help your wife. So I joined formally as CEO. The first thing I did was file all the ideas into a patent. And then thanks to some of my friends, I was able to take the biological solutions, push it towards things that I'm comfortable with, which is microfluidics. Got Onop, another Gularis student. He actually helped build the first prototype. All along, Priya kept telling, hey, there's a new professor moving from Harvard, Sunita Nagraj. You need to meet her. She's she's very good in this area, especially microfluidics CDC. I told Priya, I'm the CDC expert. I'm the microfluidics expert. Why do you need another chemical engineer? So she said, you got to meet her. So that's what I did, and I'm lucky to have got Sunita on board as an advisor. Incredibly, I think her help saved us probably at least a year of doing the wrong experiments. So I think it's important to listen to others and try to get as many experts helping you out. So again, the competition that Priya and her team went in 2010, Exeter, Michigan, actually that's where they had a sea change in their own attitudes towards business. We actually applied for that, and then we became the semi-finalist. And then we had a couple of rounds we had to go through in like 3 days for that competition. And then I was preparing all my pitches along with my team, and there are oftentimes I was not doing a good job, and then my wife and the other two made me practice about 30 times until I got it right. And then when the competitions came, we essentially went through all the rounds, and at the end, when the final was 10 companies giving the pitch for 10 minutes, I had the passion of the entire team on my head, and I had to give my best performance ever. And other people said at the end of the event that de Novo Sciences was head and shoulders above all the other competitors, and we won the $500,000. That was the time we really started building the company out, because when I joined as CEO, I gave myself 6 months. If I can't raise money, this is not, the HandyLab, acquisition and whatever money I got was really a struggle, 9 years of, working really hard, and then I wouldn't give away, that money unless I know for sure I was going to get some benefits out of it. So I had given myself 6 months to raise some capital, and within 4 months we raised $500,000, and that actually helped me get some angel money, $1.75 million to coalesce the company, get started. So again, I think it was a really good learning experience, trying things out, and I'm lucky to have had the experience from the first company to get some things right in this one. And if you look at all metrics, we are doing great. So we wanted to make sure we are out in front of the customers very quickly. In 2013 AACR, we launched our prototype. Even though the prototype looked very janky, it's like a pizza box, we still went ahead and went to the market because we want to make sure we want to be in sync with the market. We want to know what problems they have and our solution will fit or not. So this was a huge milestone. 2011, end of 2000, we got money, and in April 2013, we went with our prototype to the conference. We refined it, late last year, 2014. We got it all right. We actually made a couple of instrument sales. Now I'm actively engaged with distributors across the world. I have a distributor in China, Japan, not China. We're talking to someone. India, Japan, Korea, Israel, Qatar, and obviously there are a lot of discussion in the US. So I think this is a company that's trying it out in the marketplace and trying to get it right. Again, I like to always end with some key lessons. Again, you need a team. You cannot do great things alone. You need a team of skill sets that complement each other. And then commitment and passion can overcome any obstacles. You don't have to have the right experience. You can learn it if you are committed and passionate about it. Milestones are critical. You have to have some really well-defined milestones where the outcome of the milestones should be two ways. If you achieve it, it'll elevate you to the next level. If you do not achieve it, you should die, because there's no dragging yourself to the next milestone. It's too tiring. And then mentoring and nurturing of young entrepreneurs can create great unexpected results. As I'm learning, becoming more and more experienced, I am becoming more and more resistant to I know too many of the pitfalls, so I'm not willing to take that risk. So the best way to alleviate that is to work with young, unadulterated minds, because they don't know about what the pitfalls out there are. When they say, I would like to do this, I think sometimes it's good to go with them, because as an experienced person, you are naturally becoming more and more risk averse. So it's good to work with young people. And then every team needs to find out its own pathway, own de novo pathway, how to chalk its own course through the commercialization world. And then, ask for help, and it surely will get help. You cannot say, oh, I'll figure this out myself. If you don't know, if it's taking too long, you just have to, ask for help. And then I think, there are a lot of people that helps you create some unique product that helps the world. I mean, there are numerous people. I think you have to be genuinely thankful to all of them for, giving you the chance to get to where you are. And I think good things will happen if you are always thankful. That's all.
The way that our polymer blends are made is that we take two polymers in solution. That means they're mixed in a liquid, and then we drop that onto a substrate, so onto like a flat piece of silicon or glass.